Hello biologists, welcome back to Bioscience. This lecture is all about membrane permeability and membrane transport. So now that we've learned about the plasma membrane, we're going to talk about what stuff can get through and how. So let's title our notes membrane permeability and membrane transport. Membrane permeability and transport. So let's start first with how different types of molecules can get past. So small molecules can get through the membrane via diffusion. Diffusion is the movement of small molecules from a high concentration to a low concentration. So diffusion, that's the movement of small molecules from a high concentration to a low concentration. So from a high concentration then to a low concentration. And a really classic example of this is like if you were to add some dye or some like a little sugar packet to a cup of water. So here's my cup of water, all full of water here. And let's say my powdered drink of choice is red Kool-Aid. I think it's so delicious. It reminds me of being a kid. So let's add some red Kool-Aid into the bottom of my cup here. And if I walked away from this cup, I didn't stir up my little, however many little molecules, let's see, 1, 2, 3, 4, 5, my little 6 molecules or 6 little granules of red Kool-Aid. If I walked away from this cup, I would eventually come back and that whole cup would be red Kool-Aid. Really diluted, but still delicious, right? And so if I were to walk away, then what would happen is I'd come back later, and apparently my cup is bigger now, and that's fine. So I had 6 little things of red Kool-Aid. So all of those 6 red dots of red Kool-Aid would be equally distributed throughout my cup, and my cup would then appear all red, right? And so this is called, so this would be a high concentration here. So if I zoom in, so this would be my high concentration. High concentration, and notice that there's like no red molecules everywhere else in the cup. So everywhere else, that's the low concentration. So everywhere else is the low concentration. And so my molecules, my red molecules, they want to move from a high concentration to a low concentration. And this proceeds until we're done, until those molecules are equally distributed. So nowhere has a high concentration and nowhere has a low concentration. This here is called equilibrium. Till everything is equally distributed. Equilibrium. So that's diffusion. And diffusion can happen in a couple of different ways. We call this process that we just drew, we call this process simple diffusion. Diffusion can be simple or it can be facilitated. So let's say simple diffusion is molecules going directly across the membrane from a high concentration to a low concentration. Go directly across the plasma membrane, PM, from a high concentration, which I'll just abbreviate to H, to a low concentration. And this proceeds until equilibrium is reached. So that's small molecules, simple diffusion. But some molecules have properties that make it such that we need a protein facilitator, a helper, and that's called facilitated diffusion. Facilitated diffusion is when small molecules, they need a specific protein. So they use a specific protein to pass through the membrane, So if I were to have a plasma membrane here, what would happen is, so I'd have a couple of phosphate heads, fatty acid tails, and remember it's in a bilayer, so I have phosphate heads, fatty acid tails, and then I'd have this big old protein that I'll draw in purple because I think purple sounds fun. So here's a protein, and I'll just color that in. So there's my protein that's going to help whatever molecule get past. And then, you know, the membrane, it's an integrated membrane protein because it's integrated into that membrane. Okay, so here's my plasma membrane. This purple guy, that's my specific protein to allow whatever specific molecule go through. So I don't know, let's say calcium. So calcium can then pass through and then to the other side of the cell. And only calcium can get through that because every protein is specific to a certain type of molecule when it's used in this way. Facilitated diffusion, you need a protein helper, a protein facilitator. And still going from high to low. And just a note about concentrations. So when we say, so let's just note about concentrations across the membrane. So when we say that something is, so here's my plasma membrane that I'm just drawing as a line. If something is going from a high concentration to a low concentration across the membrane, we say that we're going with the concentration gradient. Gradient is like a difference between two things, and it's kind of fuzzy there in the middle, like a sunset. All those colors changing to other colors, that's a gradient. So we can say there's a concentration gradient. And so when we go from high to low, we say that's with the concentration gradient. But when we go the opposite direction, still we have high, still we have low, but when we're going from low to high, that's against the concentration gradient. The concentration gradient. Because you're going backwards, so you're going against, you're fighting that current. So if you're going from high to low, that's with the concentration gradient or with the gradient, and then going against the concentration gradient is going from low to high, that's going backwards. That brings us to our next point also because, so diffusion, let me bring up our definition here. Diffusion is the movement of small molecules from a high concentration to a low concentration with the concentration gradient, right? We have a special kind of diffusion. A special kind of diffusion is called osmosis. Osmosis. Osmosis is the diffusion of water, H2O. So again, going from a high concentration to a low concentration, just like that. From a high concentration to a low concentration. And so when we're talking about osmosis, we talk about the movement of water because water is always going to go where water is lowest because that's going from a high concentration to a low concentration. And so let me draw some stuff that we're going to talk about. So we think about cells and osmosis a simplified way is to think that if we were to drop one cell in a little cup of water, so here's my one cell inside my cup of water. And so there's some things we need to talk about here. So if this is my cell, oops. So if this is my cell, the fluid inside that cell, that fluid is called the intracellular fluid. And the shortened version of that is ICF. The shortened version of that is ICF. And then everything outside of the cell is what we call extracellular. So we call this the extracellular, because extra means without, extracellular fluid. And we call this the ECF. That just means the stuff outside of the cell. And then there's a couple of other things that we need to think about too, is that we think about the concentration of stuff, and that stuff is solutes inside of the ECF versus the ECF. So let's talk about this. So osmosis, that's the diffusion of water from a high concentration to a low concentration, but we also have this other related term called tonicity. Tonicity. So tonicity is a quality of a solution. A solution is like water with stuff inside. So a quality of a solution. A solution is just water with stuff inside, like salt or red Kool-Aid or whatever, that affect the shape or the tone of a cell. So that's tonicity. Okay. That is tonicity. So it's a quality of a solution, and we use tonicity. We're always using tonicity as a comparative term, which means that we're comparing solute concentrations in the ICF, CF versus the ECF. Okay, so tonicity is a comparing word that we use to compare solute concentrations within the cell and without the cell, ICF to the ECF. So let's draw, I have three scenarios about tonicity. So I have three scenarios. And in those three scenarios, I have 3 cups of water with 3 cells in it. And all of this comes back to tonicity, so keep that in your brain as we go. So 3 scenarios, I have a cup of water, and another cup of water, and another cup of water. And within that cup of water, I have a cell. Any cell, a red blood cell, doesn't matter. And the first situation that I have is called an let's zoom in actually, isotonic solution. I notice that I'm calling it a solution. I'm not saying the cell is isotonic. I'm saying that the solution is isotonic. And what that means is that when we're comparing the concentrations in the ICF to the ECF, when I'm comparing the ECF to the ICF, those concentrations of water are equal. So the isotonic solutions means that the ECF concentration, and shorthand for concentration, just so you know, are like brackets. The ECF concentration of solutes is equal to the ICF concentration of solutes. Things like salt. Like I could say sodium chloride, the concentration across that membrane is equal, which means that the water concentration is also equal. So water still moves across the membrane, but the amount that the water is moving across the membrane kind of cancels itself out. So what we say is that there's no net movement of water. So no net movement of water. That's an isotonic solution. So no net movement of water. But if we were to change that situation and in something called a hypertonic solution, a hypertonic solution, so hyper means like too much. So a hypertonic solution means that in the ECF and the ICF, it means that ECF, that solute concentration is higher compared to inside of the cell. So we say that the ECF solute concentration is greater than that of the cell ICF. What this tells us is that there's less water where there's more solutes. Let me explain what I mean. So let's pretend that we have a bunch of these red molecules outside of the cell here. We have just a bunch of them. Versus inside the cell, maybe we only have one. Let's say we have two inside of the cell. What that means is that the water inside of the cell is more diluted compared to outside of the cell. And outside of the cell in the ECF, there's more solutes. And so water kind of wants to go around diluting everything. That's what water wants to do. So water wants to go in and it wants to help, outside of the cell and say, okay, you need to be more diluted outside of the cell. So water is going to leave the cell in order to try to dilute that solution in the ECF. So water is going to leave the cell because water is going from a high concentration to a low concentration. There's lots of water inside the cell, less water outside the cell. And there's less water outside the cell because there's more solutes. So a good mnemonic device to remember hypertonic solutions is that water follows solutes. Water is always going to go where there's more solutes because it's trying to dilute everything. So back here, water leaves cell. And when water leaves cell, it kind of loses some of all of the, stuff that's holding it together. And so the cell kind of shrivels up like a raisin. It loses some of its like really nice qualities, keeping it nice and round. So it goes from round to kind of like this raisin shape. And that's called, that cell would be called crenated. Crenated means like shriveled up like a raisin. So that's hypertonic solutions. Let's move on to hypotonic solutions. That's the last scenario we have. Hypotonic solution. So we still have the ICF and the ECF. The solute concentration outside of the cell is actually lower than that inside of the cell. So when we're talking about ECF versus ICF, the ECF is less than the ICF. Hypo means like less than or below. So hypotonic means the solutes outside the cell are lower. And that means that water is actually higher. So let me draw. So that means like maybe we have two solutes outside and a bunch inside. And remember, water is going to follow those solutes, so water is going to go into the cell. Water goes into the cell, so H2O enters the cell. And when water enters the cell, it can get really full of water. And when it gets really full of water, that water pushes on that membrane pretty hard, and that causes an increase in what we call osmotic pressure. Because the force of osmosis is strong, so it's pushing on the outside of that membrane pretty hard, that water being filled up in that cell. And sometimes when that osmotic pressure gets really high, that cell can explode or break, and we call that lysis. So if extreme, this can lead to lysis, or the cell being lysed. Lysis means exploding or breaking up into pieces. So those are different properties of solutions and how they can influence the shape or the tone of that cell. So we have Isotonic solutions, no net change of water movement across the membrane because those solute concentrations are equal across the membrane. If we're in a hypertonic solution, that cell has more solutes outside of it, so water is motivated to leave because water is always trying to dilute stuff. Water is going to follow those solutes. And then lastly, in a hypotonic solution, water is going to want to enter that cell, and sometimes that can lead to lysis. So that's tonicity and osmosis. The last phrase about diffusion that we need to talk about, because osmosis and tonicity, those are special types. Osmosis is a special type of diffusion, but let's chat for the last thing. So we have passive diffusion, but we can also have active diffusion. Active diffusion. Active diffusion, remember we were talking about against the concentration gradient, right? So that means we're going backwards. So movement of molecules against the concentration gradient. Concentration gradient. And that's going from a low concentration, so high to low, from a low concentration to a high concentration. And because we're going backwards, we're kind of like fighting it, this requires energy input. Requires energy in the form of ATP. That's cellular energy. Active diffusion. Okay, so that is the transport of small molecules, mostly diffusion. If it's passive, we're going high concentration to low, and if it's active diffusion, that's when we're going against the concentration gradient from low concentration to high. That's how small molecules move across the membrane. But if it's bigger stuff, bigger molecules, or even like bacteria and stuff, then there's a whole different thing. And this is what gets us into membrane more things about membrane transport. So this is membrane transport of large stuff. That stuff can be large molecules or bacteria or whatever. So membrane transport of big stuff. So we have two ways that we can transport stuff. We can either bring stuff into the cell or we can bring stuff out of the cell. So we can bring stuff into the cell or we can bring stuff out of the cell. If we bring stuff into the cell, that process is called endocytosis. Endo means into, cyte means cell, osis is a process, so it's the process of bringing stuff into a cell, endocytosis. And taking stuff out of the cell, that process is called exocytosis. Exo means like to exit, cyte means cell, osis is a process, so it's the process of exiting stuff from the cell. So I'll start with exocytosis because, I don't know, I just want to. So we'll start with exocytosis. So exocytosis, we have a cell. There's my cell. And that cell, let's draw this better, huh? Let's make sure we're not filling the color. There we go. Okay, so I have my cell. And in my cell, I've made some product. And I have this thing that I make. It's like Amazon Prime of the cell. It's called a vesicle. And that vesicle, I have some, I don't know, blue molecules in it. Boop, boop, boop. And I want that vesicle, so that, let me label that vesicle actually. That's a vesicle. Vesicle. Vesicles are how we transport stuff in the cell. This is a cell. So what happens is that vesicle also has a membrane, so that membrane will fuse with the plasma membrane. So let's draw this. So that vesicle fuses with the plasma membrane. So it kind of goes like this. Oops. eraser, that vesicle fuses with the plasma membrane, and I still have 3 little blue molecules. And then that blue stuff just gets to leave the cell. That's kind of nice. So then again, then that blue molecule gets to leave the cell. Pretend that's a cell. And then 1, 2, 3. So that's exocytosis, the process of something leaving a cell. Endocytosis is the reverse, bringing something into the cell. We have 2 types of endocytosis, 2 types. Oops, let me use black. We have pinocytosis. Pinocytosis is like cell drinking. The cell is going to sample that fluid outside of itself. So pinocytosis, that's cell drinking. It's pretty much the opposite of exocytosis. So just kind of reverse this. That's pinocytosis. The other thing is, the other type is called phagocytosis, PH, phagocytosis. Phagocytosis is when you're going to eat something, to phagocytize it, to eat it up, break it up into pieces, and spit it out. This is something that our white blood cells are doing all the time, like bacteria, little dust particles, that kind of thing. So phagocytosis is bringing something into the cell to ingest it, to break it up into small pieces. And so how this looks like is, okay, so I have a cell. And what my cell, well, let's start with the foreign thing, actually. Let's use red, because red is, so let's say I have my little bacteria here. I have a little bacteria, and I don't want that bacteria around my cell. So what my cell does is, so here's my cell. What the cell does is it kind of reaches up. The plasma membrane does. And these little feet, these feet-like projections of the plasma membrane are called pseudopods. Pseudopods. Pseudo means fake, pods are feet, so they're little fake feet of the cell because the cell's reaching up to kind of grab and engulf that bacteria or whatever it is. So we form pseudopods, and then we've engulfed that thing now inside of the cell. So then I have a little vesicle with my little red bacteria in it. And then we fuse with the lysosome. So this is lysosome. The lysosome is an organelle that has a bunch of like digestive enzymes to kind of break everything up into little teeny tiny pieces. And so that lysosome will join with that vesicle containing my bacteria. And what it does is it forms, it's called the phagolysosome. That's called the phagolysosome. What happens is that little red bacteria just gets like broken up into these little teeny tiny pieces. Because in the lysosome, there's all these like little digestive enzymes. That's these little green guys I'm drawing. Digestive enzymes, they kind of just break everything up. And then, well, now we have those bits and pieces, we want to get rid of them. So how do we get rid of them? Then we do exocytosis. So it's kind of like 2 types, or 2 things happening at once. So now, all those little pieces of the red bacteria, they exit the cell because they do exocytosis. And now we have one little happy cell. Now our cell is just happy again. So this process here, that's exocytosis. The same process here, same thing. So those are the different types of ways that we can transport large things in the cell. We can bring stuff into the cell, endocytosis. Either we can do cell drinking, that's pinocytosis, or we can do phagocytosis, bringing in a bacteria or something, ingesting it using our lysosome, and then exocytosing all of the discarded bits. And that process, speaking of exocytosis, is making stuff inside the cell leave the cell by fusing a vesicle to the plasma membrane to then release its contents. That's all we have going on for this video, and I'll see you in the next one.